Hi everyone, welcome to case study number 2. And similar to case study 1, I hope you had a chance to download the input FASTA file for this case study and run it through SeqScreen on your own. If you weren't able to run SeqScreen, I hope that you were able to download the pre-computed outputs and practice interpreting those results on your own. I selected a publicly available cloning vector sequence for this particular case study because I believe it's representative of a common challenge that I've seen with biosecurity screening of viral sequences, particularly viral vector sequences, if they've been derived from a pathogenic virus. So we're going to talk about that more. But before we do, I wanted to point out that within this example, I did provide you with the name of the cloning vector sequence and the GenBank ID that you could use to look it up and learn more information about it before you analyze the results. And that is completely fine. As a bioengineer, you are going to be using many different cloning vectors in your work. And I think in just about every case, you will have lots of information about that cloning vector. And I encourage you to learn about that and consider it and look at the results of that cloning vector through your biosecurity pipeline so you understand what the expected results are going to be as you begin to modify sequences and use it. You can then interpret results within that context. So if we go to this cloning vector GenBank ID here, we see that this is a cloning vector sequence from EV71, and it's meant to, so here's EV71, polyprotein 1, and then a particular peptide within polyprotein 1 of the EV71 virus. And its purpose was to be a candidate dual vaccine for both EV71 and CVA16. And if you don't know what all those letters and numbers mean, that's okay. Basically, these are two viruses that are involved in hand, foot, and mouth disease in children. So they cause this disease in children, and the purpose of this cloning vector is to try to help develop a vaccine against this particular disease. And it's very common in vaccine development to only use part of a pathogenic viral sequence in the development of that vaccine. The reason for that is that we want your immune system to be able to recognize that particular virus by looking at part of it, but not infect individuals with the entire pathogenic virus within the vaccine. So we're looking for something that'll be effective in generating immunity without being pathogenic and making someone sick. And that's exactly what this is. So this cloning vector itself is not pathogenic. It is safe. It's only using part of the sequence of these viruses, specifically the EV71 virus. And to give you some additional context for that, this is a publication that had a nice figure, so I grabbed that. But just to give them, kudos and references, here's the publication that we're looking at, and we are looking at polyprotein 1 of the EV71 virus, specifically a region of peptides in this area of polyprotein 1. So there you have it. This is a portion of a pathogenic virus. But the big question is, what does this look like in a SeqScreen output file? So what could we expect to see when we're looking at a viral polyprotein through the SeqScreen pipeline? So glad you asked. Let's take a look. So this is my output file that you can download on our GitHub repo within the case study 2 directory. And once again, I have a number of different tabs in this spreadsheet. Those tabs are corresponding to different SeqScreen output formats, as well as different intermediate files to give you more information about the alignments that were done to generate these outputs. In fast mode, we see there is a single hit to a protein that's called genome polyprotein. But interestingly, the organism is not a virus, and that is because, unlike in case study 1, where centrifuge did not return a result for the input sequence, here centrifuge did return a result, and that particular 1405 NCBI tax ID corresponds to this bacillus species. But when we look at the confidence for it, it's a very low confidence. So I think this is likely due to differences in the database composition between centrifuge and diamond, but centrifuge did not find a very good match to this sequence. However, since it reported something, that is what automatically got reported and populated in the fast mode results. We can see in the diamond multi-tax ID field that there are a number of other tax IDs reported by diamond. And when we look at those in more detail, we see that, so these are the diamond results, and there are a number of top hits that have similar bit scores and e-values, and those all are genome polyproteins from enterovirus A71. So this definitely looks like enterovirus A71 from these confidence values and these results compared to the centrifuge result. You may be asking, well, why did SeqScreen report the centrifuge result then, if that was lower confidence and diamond was higher confidence? And the reason can be found in the SeqScreen documentation. So if you remember, within the 3 different modes of SeqScreen, we have lots of different tools running. Within sensitive mode, you can optionally run BLASTN in addition to BLASTX. Within fast mode, we run both diamond and centrifuge. And then within ONT mode, we do just run diamond, but we have a couple different diamond databases. One is a higher quality database with higher quality functional annotations. That's similar to what we use in fast mode and sensitive mode. But then we also have a lower quality database. If there's no hits to that first one in ONT mode, we'll go to the other one to increase sensitivity of results. All that to say, there's a lot going on in SeqScreen. Lots of different tools are being run, and the developers needed to come up with a way to decide which tool to go with in reporting the final results if they both gave you different results, which one to select. And when we go to the taxonomic identification workflow wiki page here in the SeqScreen documentation, we can get more information about all things taxonomic identification workflow related. There's a handy table here that'll tell you what databases are being used with the different tools. There's also some asterisks here that give more detail about how the databases were constructed, where you can find them, all kinds of details here. What I want to draw your attention to, though, is at the very end of the documentation, there's some discussion about the final taxonomic logic. And for sensitive mode, when both BLASTN and BLASTX return tax IDs, BLASTN is given preference. So BLASTN results will be listed first before BLASTX in the taxonomic field. And then in fast mode, centrifuge is given preference. So the only time that diamond results are listed in the single tax ID field or the multi-tax ID confidence field is when centrifuge does not return a result. And in case study 1, we saw centrifuge not return a result, so we were looking at the diamond multi-tax ID confidences. In case study 2, centrifuge did return a result, and even though it was low confidence, it was something, so it was output in that field. But this goes back to just knowing how to interpret the outputs. I would definitely recommend that you look at the confidence values for the tax ID assignments, and if centrifuge happens to be low, then take a look at the diamond ones and see what those look like. That's what I just did in this example. All right. One other point is that I don't know that one tool always gives you the correct answer. Perhaps that's a dirty secret in bioinformatics, that different tools will perform well in different scenarios. And sometimes centrifuge gives you a great result, and sometimes diamond gives you a better result. And we needed to pick a decision path here to consistently have our software give answers in a uniform way. But we included all these different results in here so that you would have more information as a user, so that if one tool did not give you a very confident result, you could always go back and look at the other one. So that's the high level rationale here. And hopefully that helps clarify things. Okay, so before we move on from these fast mode results, I did want to go through and just talk about the functional annotations that were assigned to the UniProt ID. So even though we just focused a lot on taxonomic classification and discussing why this bacillus species is showing up here, the actual UniProt ID is from the virus EV71. And it's a genome polyprotein, which means that there are many smaller functional units within this polyprotein that are actually used for different functional purposes for the viral processes. And that is a point of confusion and challenge within databases right now as we try to do functional annotations and assignments and look at biosecurity through a functional lens. Because if the best match for a protein for your particular sequence is a viral polyprotein, that means it's going to have a really long list of functions, but not all of those functions actually correspond to your small sequence, because now we're looking at the entire polyprotein and listing all of those possible functions rather than those that are just specific to your short sequence. And that has to do with the way that these proteins are organized within the database. I think this is definitely an area where more research and focus could be done in breaking up these polyproteins into smaller functional units and just organizing our databases and reporting a little bit differently in the future. But for right now, this is a challenge and it's something to be aware of, so that if you get a result like this, where you're getting a genome polyprotein and it's got all kinds of fun socks assigned to it, and that raises alarm bells in your mind that this could be a pathogenic protein, just keep in mind that this is a polyprotein and not all of these functions necessarily correspond to the short sequence that you were analyzing. So the functions that we're getting returned here are corresponding to the whole viral polyprotein. Hopefully that made sense. We're going to see a similar story with the other modes. So here we have sensitive mode. So some differences that are worth mentioning here. So for the organism or the tax ID and the confidence that's listed accordingly, we see really high confidence here from the BLAST-N result, and it's assigned to the exact cloning vector that we expected it to be. So BLAST-N is doing exactly what it should here. We're blasting against the NT database, and it's finding a perfect match to the cloning vector, which is exactly what our input sequence was. So good job, BLAST-N, you did it. You got the right answer. And we see that with the confidence value, that this is a very confident answer, and there's only one tax ID listed, so it was the best match from BLAST-N. It definitely looks like it could be concerning. I mean, in addition to all the fun socks, we also see that it could be a BSAT, so lots of concern going on here. However, I'll just reiterate again that even though it might have some similarity to something on the BSAT list, and it has a lot of fun socks assigned to it, this is not of high concern from a biosecurity standpoint, because we are looking at all the labels right now from a genome polyprotein. And this UniProt ID is once again a polyprotein from the EV71 virus. It's a different UniProt ID than the one we saw previously, but if we look at the list that sensitive mode is returning, this is the one we looked at previously. This is the one that's on top for BLASTX and sensitive mode. And when you look at the E values and the bit scores, they're all very similar. So similar story, slightly different UniProt ID in BLASTX compared to Diamond, but same take-home message here. And then with ONT mode, once again, we see the top hit being a genome polyprotein to enterovirus A71, and the UniProt ID is similar to what we were just discussing. Something I haven't mentioned is that PathGo annotations will rarely appear for viral polyproteins because our viral curation team does not bother to annotate polyproteins because of all the challenges I just mentioned. It would be much more meaningful to analyze smaller functional units and report those from a biocuration standpoint than from the entire viral polyprotein. So they don't even bother with these You will rarely get a PathGo list of terms for a polyprotein. You will, however, get that for FunSox because the machine learning algorithm will assign those. And these aren't wrong for the polyprotein, but when you interpret the results, you just have to remember that what is true for the polyprotein is not necessarily true for the smaller subset of the sequence that you were analyzing. Other things that we see in ONT mode, relatively high taxonomic confidence here. So here we're looking at the diamond result for the ONT mode, and that gives us the enterovirus A71. So whereas in fast mode, we were looking at the centrifuge result, now in ONT mode, we're looking at the diamond result, and it gives you the correct organism. And then we see a bunch of other stuff here in the ONT results, and that corresponds to other open reading frames that it's finding within this cloning vector, and it's trying to decide what other genes are present within the cloning vector sequence that was provided. It thinks it has found some different antimicrobial genes, antibiotic resistance genes, other genes just involved in normal processes of cellular metabolism. So nothing particularly concerning here, but there are things that ONT mode is picking up on that the other modes didn't, as far as other possible open reading frames present within the sequence. And then these are the diamond results for ONT mode, which looks similar to what we've talked about before, where the top hit is this one. Okay, so that was my example for a viral polyprotein result. Again, this is something that commonly happens in viruses, and that is an artifact of the way that we have organized our databases around viral polyproteins. And when we're looking for functional labels, we have to keep in mind if our functional label is coming from a polyprotein, it's not specific likely to the small subset of a sequence that we were analyzing. It's looking at the entire universe of possible functions for that virus. Okay, great. Thank you so much. And I will see you at the next case study.